So good morning, everybody. My name is Leslie Mitchell, and I'm a postdoc in the BUCA group here in New York. I will put my talk on. And so you'll notice as these slides come up, I've changed and sort of simplified the talk title just a wee bit from what's listed in the program. It's going to go? Okay. To a slightly simpler title that covers something we've been working on actively in the BUCA lab, delivering what we call big DNA to mammalian cells. And so I think what we're here all today, at this meeting to talk about is the fact that we're no longer limited to the study of cells that are the product of natural evolution. We're now in a position to write and edit genomes in a way that we can build new functions and direct growth of cells with new functions by design. And so when I think about this burgeoning field of synthetic genomics, I sort of break it down into two sort of big classes of thinking about editing and writing genomes. Those are the two classes. And this was an analogy from Joel Bader, who may or may not be here yet. And he was talking about this in the context of the SC2 project, this idea that in some cases we want to go in and make some very focused edits, much like in this Word document, paragraph 1, where maybe there was a typo or a word that was incorrect. You can go in and make that very focused edit, and it makes sense to make that change in the context of that paragraph. In other cases, like paragraph 2, there's a million edits to make, and it almost makes more sense. In fact, in the case of SC2, for instance, it makes more sense to go back, design it, and write it from scratch in a way that then we need to think about how to deliver it to cells. And so for SC2, that meant edits that were densely spaced, approximately 400 bases between clusters of edits. And so we built that from scratch. So starting to think about this overview of de novo design, and I'm not talking about the design here, but how to get that from the design and the synthesized fragments or whatever other starting materials you have, which might include BACs, YACs, or PCR amplicons derived from your source material. You can start thinking about, let's see if this works, this sort of layer of the workflow that involves assembly and editing, and you can use cells for those activities and all sorts of editing tools to build your big DNA of interest, which can then be either delivered to your destination cells by conjugation from E. coli, and I know that's not a particularly efficient process, cell fusion, which Alina talked about yesterday, or via purified DNA and just standard transfection protocols. And so a lot of these activities are now underway in the BUCA lab. For the SC2 project, of course, the assembly and delivery are all part and parcel in the sense that our delivery overwrote the existing DNA, and the activity of homologous recombination in yeast is so efficient that the delivery wasn't a big challenge, but we see this as a major challenge for GP Write. So just to break down very, very simply these challenges, it comes in right up front when if you're working with purified DNA in the handling in vitro. I hope you can all see this cloud of DNA that's precipitated in this ethanol solution of a 100 kb construct that I purified by cesium chloride last week. How exciting. As Alina was talking about those old school methods, they're coming back to life. So in the act of purifying bigger DNA, it can get sheared in a way that the intact transfer into cells is limiting. Once it's in the cells, it needs to be delivered into the nucleus. There's a number of pathways of naked double-stranded DNA that can activate antiviral pathways. And then, of course, the targeted delivery or site-specific delivery. And I'll talk about some of these strategies that we're thinking about in the BUCA lab. So we're in sort of not early stages, but like not quite ready for prime time. So I'm going to highlight some of some of the great literature that's out there to give you guys an idea of the activities that we feel are important for delivering big DNA. So as Alina highlighted yesterday, this is work from her and David Brown, was published earlier this year in NAR. This idea of delivering big DNA via spheroplast-mediated cell fusion, which is just an event requiring spheroplasting of the yeast cells, fusing it with the mammalian cells, and by virtue of that fusion, delivering the contents of the yeast cell into the mammalian system. And so David Brown et al. I think I'd really recommend, if you're interested in this type of work, to go back and read this paper. Are reporting a design of experiments kind of workflow that allows you to optimize based on the destination cell type. And they're showing efficiencies for delivery sort of independent of length scale on the order of 1 in 1,000 cells. So the use of this spheroplast-mediated cell fusion is actually long-standing. We've delivered in the past megabases of DNA in untargeted integration to mouse ES cells and cloned the T cell receptor, which is a 1.2 megabase YAC intact. That was untargeted. Another really important approach to think about is the targeted delivery of DNA. And I think one way we're thinking about this is the fact that genomes can indeed capture linear DNA that's present in the nucleus, regardless of homology. So this idea that you can deliver there's two approaches that are shown here from two different groups. If your double-strand cut site exists in your vector as well as the genome, you deliver that circular DNA, and it's a relatively efficient process to deliver that entire vector into that cut site via a single double-strand break, either CRISPR or TALENs in these cases. Site-specific recombination is another really important strategy that we're working on, and so this is like the precise DNA cleavage and ligation without loss of nucleotides. There's a number of existing heterotypic site-specific recombination systems that are listed here. All of these require a pre-existing landing pad to be engineered into the cell. And so this is often thought of for delivery of big DNA in the combination of recombination-mediated cassette exchange. And I think one of the best examples in the literature of this is from Doug Higgs's group at Oxford, where they've delivered, they call their process recombinase-mediated genomic replacements. And they've replaced the mouse alpha globin locus with a human alpha globin locus. So they engineered a back to have recombinase sites with a selection system and engineered the native locus to generate a landing pad. And this slide shows exactly how they did that, and it's super complicated, so I'm just going to highlight the actually important parts for the delivery. So into the mouse landing pad, they've inserted half of an HPRT gene, which is a selectable marker, flanked, and there's a LOX site, a heterotypic LOX 511, I believe, site on the other end with a thymidine kinase anti-selection resident gene. On the back, they've engineered it in a way that it can be delivered to recapitulate the HPRT gene for hat selection and overwrite the resident thymidine kinase for ganciclovir anti-selection. And then they can pop out that HPRT gene using FLIP. So this is a really nice strategy for the delivery of big DNA. And we're coming up with sort of takes on that approach. And the way we see all this fitting together is represented back to the initial diagram, is building out largely in yeast, and I'll talk about that later in the Foundry session, 100 to megabase size molecules of DNA in yeast. Then that can be transferred via these fusion protocols directly into the mammalian system, where we've already pre-engineered and characterized landing pads that are integrated by these double-stranded break, and then integrating the landing pads via MMEJ or NHEJ. And then using, we're going to call it, and we're not going to unveil it today, unfortunately, but approach called BigIn, which is a take on recombinase-mediated cassette exchange to introduce this engineered DNA into the existing landing pad. So this allows for editing and engineering of the specific DNA sequence in these assembly and editing sort of in this platform, and then the seamless delivery without having to go in vitro to deliver big DNA into the mammalian systems. And so with that, I'll wrap up. Here's a list of people involved in all of this work. The team Big DNA, which involves group members of the Buca lab as well as Matt Morano's lab. I'll talk about the efforts for our automation suite later today, and the entire Buca lab. A number of people are thinking about the design and construction of synthetic chromosomes and genomes, of course. So if you have any questions, I'd be happy to take them. Thank you.